communicate with you. So, first question, why? What is diagnostics? I just give the question to you. What is diagnostics? Just to define the cause of disease, yes. And further on, to characteristic particular species, genesis, and phenomena as an overall expression. Further on, concerned with the diagnosis, illness or problems. That is what we try to identify and try to cure in our further progress. That is the diagnostic tool. And finally, distinctive and indicating nature of an illness. Of an illness you should put, in fact, in brackets, because damage on plants is not always an illness. I will show you there's various symptoms that are not illnesses, but factors from other causes. Why do we need diagnostics? In principle, you could say, okay, a healthy plant doesn't get sick, and all this diagnostic stuff, forget it. In a part, Heisam also already, well, let me put it that way, glorified aquaponics, as plants don't get sick in aquaponic system. Well, let's see. First, we have to identify a situation as abnormal. What is abnormal? Or on the other hand, when does a plant grow normally? In what system? A plant in soil, somewhere outside, might express a different phenotype as a plant in a greenhouse or a plant in a hydroponic or aquaponic system. So if we find these abnormalities, then we have to find the reason, the cause for those abnormalities. I deliberately choose the word abnormality and not illness or disease, just as I explained earlier. And then define a mode of action to act in that specific case, and if possible, define a mode of action in the future, in future cases. Preferably in an early stage of this abnormality when you discover it. And then select adequate modes of action to counteract this abnormality. In fact, we try to reduce the abnormal symptoms. In case of an illness, we try to fight the pathogen or whatsoever, hoping that at least the damage on our crop, on our plant, will not increase. Is it possible to decrease damage on a plant?
Depending on the stage of growth.
Or depending on?
What is the cause of our problem?
Yes, but mainly depending on what you wish to harvest from your plant. Because, for instance, a tomato, diseased tomato leaves don't look good. They reduce photosynthesis and they reduce the overall productivity, but they do not damage your crop, your tomato. Lettuce, it's different. A brown leaf on a lettuce plant is brown. You'll never get it green. Okay, painted green, but not naturally. So once a plant is damaged, you need to be as early as possible to defend your plant, to defend your crop, to fight any loss in your crop. So what means of diagnostics do we have? First, visual, optical means. We look at our plants, we see something that we define as abnormal, and then we say this plant is, what is it? Is it ill? Is it overwatered? Is it heat stressed? What is it? What is another means of diagnostic? Visual. In part, we discussed it this morning. What is another symptom, maybe, of health or disease of a plant? What other ways of finding do we have? If we're talking about yield, about crop, we don't only want to market it, the one or the other has to eat it. We have organoleptic means of diagnosis. We can touch, we can feel, we can taste, we can smell. All those are means to diagnose a abnormality, again. And we have technical means. What kind of technical means, for instance? Some kind of sophisticated apparatus or what? Pardon?
I don't know, maybe ELISA or PCR.
Yes, okay. But first do the first step.
Or
That's the
some
seventh.
biological methods to find.
Okay, but think of the first one. Yes, some optical means, technical means to increase your optical abilities. Microscopes, something like that. A glass. I refuse to usually, but sometimes I need it. So that is a way of technical diagnostic method. And of course, the combination of all. Usually diagnostics is a cascade. First you see, feel, smell, then you use smaller technical gear like microscopes or something like that. And if that doesn't bring you to the result, you need more sophisticated apparatus like ELISA or PCR or something like that, which I will deal about later. So, what do we need for diagnostics? First of all, we need knowledge. We can see there is something miscolored or misgrown or whatsoever. But finally, we need the knowledge, or we need to know where to look it up, what might be the cause of this damage. And then we have several stages. We have an early stage, we have a severe stage, and an end stage of damage of a disease or what. And then the other part of knowledge, what to do about it. What is the cause of the damage? Is it abiotic or a biotic cause? And which diagnostic tool to apply to finally get the answer we're looking for. What is an abiotic cause of damage on plants?
Overheat, too much water, less water,
Yes.
nutrition.
Nutrition, yes, yes, definitely. I just mentioned it. That's why I tried to ask. Sleeping, everybody. Lunch sink. Okay, and then, of course, the knowledge of remediation measures. What can we do? How do we do? When do we do? And still, we always have to keep in mind the species or the cultivar of a plant, of a crop, of a fish, and what specific, whoops, I missed the word. What typical reactions a cultivar or a species might give to any kind of remediation. For instance, if you're talking about plant protection, we have several chemical or biological agents that might kill a fungus. But all those agents affect the plant too. Nothing is without any effect or side effect. And depending on the species or even on the cultivar, this remediation method we apply might help or definitely destroy our plant. That is very important to know. The growth conditions are also very important. If you apply a remediation with the wrong conditions, too much of light, too little light, wrong temperature, also on, you can do more harm than cure. And if we then use those pesticides, again, we need to know what effect it might have on the plant, on our aiming organism course. So if we're talking about damage and symptoms, in this case, lettuce early stage, I hope I can show you this tomorrow on diseased plants. Did you find, Caroline? Something like that? Okay, at least this is an early stage, definitely of some kind of damage. This is a healthy plant. This is somehow damaged. Few days, round about a week later, we have the severe stage. The plant looks like this. And then at the end stage, we find something like that. And if you take a close look, here is just necrosis, brown material. Here might be something to see, something that might help us to identify. And in this case, you see this gray something on the plant, definitely not from the plant itself. So probably it is from the cause of damage. And this helps us to identify, in this case, the pathogen. So again, abiotic causes, up to the nutrients, biotic, virus, bacteria, fungi, and insects. I will show you a few pictures just not to leave it out of the discussion of insects, but as Haitham already said, fungi are the most important. Diagnostic tools, simple physical and or sophisticated technical. What might be simple physical? Sorry?
Removing damaged plant,
Yeah.
destroying it.
Just, pick the damaged plant out of your floating system and hoping that will be it. Or just discard the parts of the plant that are damaged. That is the most easy way. Because then you can skip those sophisticated technical for identifying. That would be the easiest and the most convenient way. But then, if that doesn't help, we need some kind of remediation methods. Again, species and cultivars, knowing, growth condition is important, pesticides, okay, symptoms. Here in this case, an example, Peronospora, subspecies, because Peronospora has various species and they're all referred to as downy mildew, but depending on the host plant, you're dealing with another fungal species. Normally, that is not of any importance, but in special cases, it might be, just as with the host plant, that the species of pathogen react different to one way or the other remediation method. So sometimes it is necessary to define the species. So this is Peronospora on cucumber. Look for the symptom. It's easy to see. It's easy to identify. This is the same pathogen on leek. Looks definitely different. And again, the same pathogen on lettuce. What now? We have a problem. What is what, which is which, and why and where? So you see in this case, it is important either to know about the symptoms of the pathogens, let us stick with pathogens right now for the fungi, and it is necessary to know where to look it up. Data banks, internet, often helps very well. But if we have abnormal symptoms that we wish to assign to a specific cause, then we have to follow a certain procedure. At least if we're not going into too sophisticated situations like PCR, because it is very expensive. It might look easier than those Koch's postulates, but the Koch's postulates are quite simple for everybody to apply. Does anybody know about those Koch's postulates? What is meant about this? No. Okay, first you start to isolate the potential pathogen. You try to bring it in culture and try to identify it from its typical means of growth or whatsoever. So you have to identify it. Identification is the second part of the Koch's postulates. The third is inoculation. Now you might ask, why should I inoculate a healthy plant with something I isolated from a diseased plant? It must be crazy, ruining my crop completely. In fact, you are right. That is the craziness of plant pathologists, because our job is to create methods, means and methods, to get plants ill. To put a disease, a specific disease, onto a plant. And then define another way of, I know how to cure it. Okay, draw your own conclusions. But further on, you have, after this inoculation, re-isolate that fungus that you have artificially put onto a healthy plant to prove that the symptoms you observed first are indeed the same after inoculation. And then, again, re-identification. So is this one you isolated first, the same you isolated second? And so are the symptoms the same? Are the isolated fungi, pathogens the same? And then you have fulfilled the Koch's postulates, and then you can be sure that your first idea, your first identification was true. How does it work? You get such a wonderful plant. I had to combine those two parts of the picture because it didn't fit in one. So you see here the diseased plant and the root system that belongs to this plant. Everybody probably will agree that this does not look healthy, neither up surface nor down the surface. Okay, what do? We have here some kind of symptom. We excise it and put it onto a petri dish. Synthetic nutrient medium, and then hope that it will grow on our medium. We have several standard mediums on which almost everything, everybody grows, be it bacteria, yeasts, fungi, or whatsoever. Might be a problem, but I skip that part of keeping your pathogen clean, because that would be another lecture of an hour. And then hoping that it grows and then maybe performs such kind of an organism. The one or the other who might be an expert and who can't read might identify this already as a botrytis culture. We're coming to that other. Okay. But we don't know, of course. Just looking at it might be difficult, might lead us to a wrong decision. So we have to identify it by looking at the typical fruiting bodies, spores, sclerotia, something like that. Everything that this thing in our on our medium displays which we can look, compare to literature, to knowledge, or whatsoever. And under a microscope, we find this. Wow. Quite typical. This looks quite good for a botrytis. Do you agree?
Yeah.
Okay. So, if we looked it up, and we find the severe stage of our plant, and find on that, you remember this grayish something on the plant, we find the same structure under the microscope like from our synthetic medium, then we can be quite sure that we're talking about botrytis. That is one way of identifying a pathogen. But I wouldn't talk about that if it would be so easy. Again, the same step. Displaying this kind of growth on our medium looks different. An idea?
Different pathogen.
Definitely. What might it be?
Rhizoctonia.
Go and have coffee. The same procedure. Again, cultivating, looking under the microscope, looking for typical fruiting bodies, looking for typical structures, and you see, damn, you don't find any. Nothing. So you go on, no fruiting bodies. So you go on, another plant of which you excise the same kind of fungus. No, in this case it's the same. But you find some other structures. Yes, sclerotia, which are not formed on the culture medium.
But you have 90 degrees branching there in Haifa.
Yes. Want to go on?
No.
Definitely, yeah, absolutely correct. But that is what I meant earlier by knowledge. You have to know that Rhizoctonia has this quite typical brick-like septation and the almost rectangular branching. That is very typical for Rhizoctonia. And it has always, usually this brownish color. And then finally, yes, if everything fits, you're right, thank you, 10 points. Then finally you have it, the collar rot, root rot, damping off, or whatsoever are the common names. And this brings, at least to me, another problem. 7 names for one and the same pathogen. Does that really help us? I'm sorry, you are no students, but usually to my students I say, learn the scientific names. And only the scientific names, because they are everywhere the same. In Hungary, in Slovakia, in Germany, in Denmark, everywhere. But if we're talking in Hungary about damping off and in Denmark of collar rot, does everybody know that that is the same pathogen? Okay, so far for this. So we have now identified 2 means to identify a pathogen. But is it really the one that caused these symptoms? This we have to find via inoculation. And this, because it is easier to do, I explain on another pathogen, on Sclerotinia. I have some already mentioned, because Sclerotinia is also a pathogen that is of high importance in hydroponic cultures and also in aquacultures. It is quite easy to identify on the sclerotia that it forms already after 5 to 6 days on a nutrient medium. But this is hard to bring onto a plant. It usually don't work. So we have to go a step in between. We cultivate this pathogen on, in this case, it's barley. And then we bring the barley onto the plant, winter oilseed rape in this case. You can see here those little kernels everywhere, hoping that weather conditions, in this case it was an open field inoculation, hoping that weather conditions will favor the disease. And if it does, then you can find a range of different symptoms in time, which then finally gives you, at least here in this stages, again, the opportunity to re-isolate, to put it back onto synthetic medium, and then to find, again, this typical structure. So re-isolation and re-identification, again, back to the culture. Great, that's it. Why do we do this?
You inoculate the seeds in that case.
We inoculated barley to make a step in between from this culture onto the plant, because this wouldn't work in the open field. It works in a greenhouse, but not in the open field. Yeah? And then it develops these symptoms. Usually it first starts from infecting the leaves, then penetrates the stem, and then completely damages the stem, and finally the plant above of this infection site wilts and breaks down.
So in between the barley seeds and the oilseed grain,
Mm-hmm.
how do you inoculate the oilseed grain?
With those barley. With the barley kernels that we first cultivated with the fungus. The fungus grows into the barley kernels. And those barley kernels you can dry or not, just as you wish, and then put it on the oilseed rape plants or on any plant. And then, if you're lucky, if the cultivar is susceptible, if the weather conditions are correct, if the fungus still is alive on your barley kernels, then you might get an infection. And yet, then you can re-isolate and then fulfill with this the Koch's postulates. First, a biochemical detection tool. It is easier, it is cheaper, it is at least as fast or faster as PCR. It's the ELISA technique, the enzyme-linked immunosorbent assay. What does that mean? Any idea? Enzyme-linked immunosorbent immune system. Yeah, something like that.
Reaction by
Uh-huh.
color is changing, but
Mhm.
depends of which ELISA you
Yes, yes. But it's off the plant. It is purely in vitro.
It's reaction, antibody, anti-gene reaction.
Yes, yes.
And,
or sometimes antibody, antigen, antibody.
okay, that's you.
Depends, yes.
Or vice versa, or competitive, or whatsoever. Yeah. So just the principles of the method. You diagnose or detect, it has begun with proteins. Now you can, in fact, detect almost every molecule that you can adhere to some kind of surface, to some kind of, well, ELISA dish, which you can then use for your antibodies as a matrix. It can be quantitative or only qualitative, but you still need high specific antibodies. And that's the problem. There are antibodies that you can purchase in diagnostic lab suppliers. You can make your own antibodies, either in chicken eggs or in rabbits or in I don't know what, mice, rats, name it. But there we go again with the time and the money. So easiest would be if you could purchase these. And for the most pathogens, for the most situations that you might find or that you might be confronted with, these antibodies are available. And the molecule that you wish to detect needs to be water soluble, because you're working in an aqueous phase. Everything that is not water soluble gives problems. It's not impossible, but it gives problems.
You can do it in organic solvent?
Usually, the ELISA plate is from plastic.
But it's only glass.
Yeah. In principle, it is possible, yes. But I never heard about that. But sophisticated methods, yes, but not in a common ELISA reader, because those dampings of the organic material wouldn't do good to your reader. You will see later on, because it is in fact a closed system also. So, okay, specifically prepared, pardon. This is the way of an ELISA. I tried to animate, but I couldn't deal with those two sides, so just let me put it one after the other. First, you have a solid phase where your antibody is coated. The solid phase is usually this plastic ELISA well. I'll show you a picture later. Then you need to block surplus binding sites, and because you want to quantify, if possible, in one step. So you have your antibody or analyte, certain quantification that fills positions in your ELISA part, and then you block with another inhomogeneous protein, usually it's bovine albumin or something like that, which is not reacting with the antibodies of plant pathogens or with the antibodies of larger molecules. Then you add usually the antibody that should be detected. That is these things. Now it depends on the method you use, if it is a direct ELISA, an indirect ELISA, if it is a sandwich ELISA or what. Then later on, there comes another antibody with an enzyme. That is what the name of this method is, enzyme-linked immunosorbent assay. Immunosorbent, immunological binding reactions, the enzyme linked to those antibodies. And then, if you add a substrate which is suitable for the enzyme to process, then you usually get a color reaction, more or less intense, depending on the concentration. And that gives you two answers. First, yes, the suspected antigen was indeed in my sample. I could detect it with this method. I get the color reaction. And on the other hand, due to the intensity of the color reaction, I can quantify. Okay, not I, but the machine can. This is this ELISA well, usually 96 wells. So in fact, you can process 96 samples at a time. Okay, sure. You need a standard, you need replicates, and so on. And then you put this well in the machine. It is shoved into the machine and then photospectrometrically analyzed. And then the machine tells you with a certain detection limit possible, it is up to 10, up to 9. So it is, help me, 10 billion, one molecule in 10 billions. So quite sensitive. Good. Let's come to the PCR. You're a PCR freak. What is PCR? PCR is the polymerase chain reaction. Chain reaction sounds good. Implicates there's something going on for itself. I don't have to do anything about it. It is not that easy. It is an enzyme-dependent technique because you need an enzyme that works for you. And that is this Taq polymerase. And that has special needs that you have to supply, otherwise it won't work. The idea is of this polymerase chain reaction, the name chain reaction already implicates, you make a huge number of copies of one molecule. In this case, it is a DNA molecule. Why do you want to know to have plenty copies? Because if you have one copy of a gene, or of a part of a gene, or a part of a DNA, you can't analyze it. You can't do anything with it. You need more. You have to have enough starting templates for the sequencing. If you come with one template, probably you won't get any kind of sequence. Or you need to bring a sack of money with it.
No, it's so cheap. You just extract DNA and sell it.
Yeah, but only if you have enough templates. Then they go into the automatized sequencer, yes. Or you need some copies for genetic engineering. This is, I wouldn't say the meaning of PCR at that time. Now it is used analytic, but there was a time when PCR was used especially for genetic engineering. So how does the PCR work? What do we have to do? First, we have to extract the DNA. And you see in brackets RNA. This technique only works with DNA. But we can use a trick to apply also RNA. That way, that we first produce a complementary DNA strand to the RNA piece that we want to analyze. I'll come to that later. Then we have a cyclic method. Usually you use about 30 to 40 cycles. A cycle means first DNA has to be denaturated. Melting, it is called. So you get single-stranded DNA. Because this is needed for the annealing phase. Annealing of the primer. You need a primer, a special molecule that is known, which anneals, which binds as complete as possible to your DNA strand, to your single DNA strand. Then you also need DNTPs. You need the nucleic acid as single parts, which then also can anneal onto the DNA, onto the single strand. And then you have the elongation phase, which is performed by the enzyme, by the Taq polymerase, which connects the primer with the base pairs that you supply into the whole analysis. And then you have a PCR product, which normally you want to analyze for length or just to identify on one way or the other to use in a gel electrophoresis to detect finally. Okay, this is such an PCR system. All that is happening in about 50 microliters of reaction mix. Yeah, thanks. So first, the DNA has to melt, has to be broken up into two single strands. Here is a standard temperature given. All those temperatures and times that are given here are standard, need to be optimized for every single procedure. Then the annealing phase, where the primer anneals to the single-stranded DNAs. And then the DNTPs, the nucleic acid parts, anneal to the DNA, and the enzyme binds it together, and then the cycle starts again. You now have 2 copies of your first DNA part, and then the second cycle brings again melting and the same situation again. For RNA, it is, well, in fact, the second step to the first. First, you have the RNA, which you have to replicate to a DNA strand, and then you can put the cDNA, the complementary DNA strand, into a PCR system. And as you can see here, every cycle doubles the number of your copies. So in the end, you have very large numbers of your product that you wish to analyze. And then usually you put it onto agarose gels. Here is the sample that you wish to analyze. DNA is charged negative, so it will go into the electric field to the positive part. Is it correct? Yeah. I always mix it up. And then you can analyze this gel for a certain DNA part which you're looking for. You have a ladder commercially available of DNA lengths. Then you can define the length of your DNA strand, and maybe you mark it on one way or the other, and then you can detect it directly on the agarose gel. Yuri, questions, remarks? Is anybody here who thinks that PCR is easier than ELISA?
I think that it's easier because you have maybe 15 minutes to prepare, then you put in machine and maybe just to prepare gel, and that's the most difficult part. And ELISA, you have more steps, and then you have 15 minutes, 10, 15 minutes, or 10.
Yeah.
For example, I worked histamine, and then you have half an hour, then you wash, then half an hour again. So it's maybe difficult, but I think that if you use that for some everyday work, ELISA is cheaper.
Yeah, I would agree. Yes, I would agree.
Because we, at our institute, we use that for some disease serological in animal, and also I mentioned for mycotoxins and for histamine, like screening tests, so it's much cheaper than some.
But only if you're talking about series of analysis. And that's the problem. Usually in your hydroponic or aquaponic system.
Yes, if you have 1 or 2 samples, it's not.
Yes.
But
Yeah,
yes,
that's the problem.
preparing PCR mix is a job about 15 to 20 minutes, but you need to get that DNA or RNA. That is a job which will
I don't scan that part.
And if you're applying PCR, you need a damned clean laboratory.
When you have just an
With Kiogen kit, you can do it in half an hour.
Yes, yes.
But with CTAP, you need about.
But you can only do the standard analysis with the Kiogen kits. Only the standard analysis. You need a special, you need a certain length of a fragment. You can't just analyze any DNA fragment with the Kiogen kit.
Yes, okay.
So.
It depends on which plant do you extract your DNA. If it has too much starch, you need to. It won't be specific enough.
Yes.
Block the filters.
Yeah.
So there's a lot of.
And then is the problem, is the DNA strongly methylated, which also brings trouble, and it sounds so easy.
Yes, yes. And then of course there are readings in gel. The DNA is colorless, and when to get this kind of picture, you need to put it in some organic.
Yeah, but
which is,
usually you use digoxygenin.
Yes, but
that is not too toxic.
Yes, but we use the ethylene bromide.
Yeah. Okay.
So
Don't do.
it's-
Don't do.
We have
cancer.
Yes, yes.
You-
It doesn't only bring a red color into the gel.
Yes.
That
It's
is,
quite,
and never use latex gloves with ethidium bromide.
no,
It is nowhere said, but it penetrates.
nitrite gloves.
Yes, yes.
So,
Please.
on this picture, so you have the sample that you analyze, so you don't know what you are looking for. So,
Yeah.
you make this migration on the gene, but what do you use as a control, or what do you compare with?
The suspected pathogen, in fact. This way, the PCR doesn't give you any information if you do not have, just like Haitham already said, if you do not have an idea, if you don't have any clue. You need a control, a positive and a negative control. In this case, let's say we're talking about Fusarium. I find this gene part in my diseased plant, and I have a gene extraction from Botrytis, Rhizoctonia, and a control Fusarium. You see, Botrytis, no match, Rhizoctonia, no match, Fusarium, match. So this will probably, not definitely, but probably be the cause. Again here, it is not definite. You always have to deal with a certain uncertainty, because it depends on the methods and on the side effects, on the temperature, on the duration time, on the magnesia concentration, on the chloride concentration in your tubes. That all can give you false results.
And it is not because you detected a certain pathogen on a plant that the pathogen is responsible each time of the disease.
Yes, yes.
For the plant.
That is the reason why in the first place for the Koch's postulates I used twice the same plant. Because of course both pathogens could be on that plant. And what information brings then an ELISA or the PCR? None, in fact. Yeah, that's the problem with these sophisticated methods. I prefer indeed the Koch's postulates. It is more complicated, but it is cheap if you know what you're doing. It is relatively fast, and it is quite secure. And you always have the pathogen for your collection.
But you can do it with all the pathogens.
Okay.
You can do it with obligated pathogens like viruses or like powdery mildews. So
Yeah. Mhm.
Phytopathology is a touchy bit.
You have to be crazy to be a phytopathologist. I said it first.
That's true.
Okay, so the last slide means thank you for your attention.